Hey everyone, welcome to Biology 1406, Biology for Science Majors, Chapter 1. Let's get started. What is Chapter 1 covering? Well, what is biology? Biology is the scientific study of life. I'm going to spend a lot of time in this chapter explaining this definition, explaining what is a scientific study, because it's not a trivial thing to understand what science is and how to conduct a proper scientific study. And then what is life, which again is not a trivial thing. Life is actually quite difficult to define, so we're going to get to this in a minute. So let's get started. What does scientific study of life mean, and what is biology? Science in general is knowledge about the universe, and humans have been trying to gather knowledge about the universe since time has began for humans. And the way we've come about learning about the universe is through a process that we've developed as humans, and that is the scientific method. A method of research with defined steps that include experiments and careful observation. Then there's like I said, defined steps. We're going to go through and talk about those steps one by one so that you understand clearly what is science and how to conduct the scientific method. Okay, but before we do, what you should realize is that science begins with observations. And so because science begins with observations, science, much of it is purely descriptive. You're describing the universe, you're gathering truths and knowledge about the universe, and then you're going to communicate those results to others. This here is the scientific method. You see, you start with an observation. I said science begins with an observation. From there, you do some background research. Why would you want to do some background research? Well, to know what's known and what's not known. You don't want to spend a bunch of time reinventing the wheel, doing experiments other people have done. So you might want to learn what is known, what is not known about that field. From there, you could formulate a question. For example, why is this observation? Why is this thing going on? What is this thing, right? And to answer your question, okay, this is the important part. You formulated a question, but to answer that question, you're going to make a hypothesis. Okay, to answer the question, you're going to make a hypothesis. And the hypothesis is a suggested explanation for an event. It's like an answer that you came up with, a possible answer that you came up with to answer your question. Now, is it the right answer? I don't know. Is it the wrong answer? I don't know. You have to test it with an experiment. So your hypothesis needs to be testable. It can't be some explanation that you can't test. That would fall outside of the realm of science. And it also needs to be falsifiable. What does that mean? That means if you test it and it doesn't turn out to be the right answer, you need to be able to nullify the hypothesis, throw out the hypothesis. Okay, and hypotheses, they actually allow you to make predictions. So for example, if your hypothesis is this is why this is going on, and you test it, well then you should be able to predict what the outcome of those tests should be. Okay, so again, science begins with an observation. You do background research to know what's known, what's not known. You formulate a question. I wonder why that's going on. And then you formulate a hypothesis. This is why that's going on. And that'll allow you to make a prediction, which you can then test. You have to be able to test if your hypothesis is the actual answer. Okay. So again, hypotheses allow you to make predictions. Those predictions you can test. You can test the predictions. You could test the predictions. You can test those hypotheses using experiments. Okay, experiments are what tests the hypothesis. And there are parts of the experiment called variables. Variables are any part of an experiment that can vary or change during the experiment. You want to have good controls in a good experiment. A control contains every feature of the experimental group except it is not given the manipulation that is hypothesized about. So if you're testing, I don't know, you're testing a drug, you want to make sure that the control group gets some kind of pill. Otherwise, if you don't swallow something, you could be susceptible to what's known as, for example, a placebo effect, where one group is given a pill, one group isn't, and the group with the pill might feel better simply because they believe they're being cured. Okay, so once you've done your experiment, you gathered data, and data are recorded observations. There are two main forms of data: qualitative data and quantitative data. Qualitative data is data that's based on descriptions rather than measurements. If you think of the famous primate researcher Jane Goodall, a lot of her results were qualitative. For example, if she's studying the primate behavior, she might note that on a particular day, a particular primate was aggressive. On a different day, it became docile. So those types of descriptions are not quantifiable, right? So they're more descriptive. That's qualitative data. Quantitative data is recorded measurements which have numerical values, right? You can organize them into tables, graphs, etc. Most data that you'll encounter is usually quantitative. Okay, so again, how does the scientific method work? You make an observation. You could do some background research to see what's known, what's not known. Formulate a question. I wonder why that's going on. And then formulate some hypotheses to try to test that question to see if it answers your question. The hypothesis is the answer to the question. But then to test whether the hypothesis is the true answer to the question, you need to formulate experiments, collect data, draw conclusions, okay? And that will allow you to test whether or not you've answered your question. And arguably the most important parts down here at the bottom, look, communicate the results to others. Others must be able to repeat those experiments. I can't stress how important this is, that other people need to be able to repeat your experiments. Science, what you should realize as science students is science is not something where people need to take your word for something. Science is not something where someone who's very intelligent had an idea and they proposed that, and now, that's a new scientific theory. Okay, that's not how science works. Science is not based on opinions or beliefs. Science is based on hard data that is gathered through experimentation and presented to others so that they could repeat the same results. If other scientists in the world, in different countries, cannot repeat your same results, it's actually not science. Okay, that falls outside of the realm of science. So if you ever wondered why something is, constitutes science and something is considered outside of the realm of science, you should just ask yourself, can I do experiments and see results, and can other people do the same experiments and see the same results? If not, it's not technically science. It does not belong in the realm of science. Okay, so it's good to understand. So let's look at the scientific method. Okay, the scientific method we've been talking about, let's look at it with this lady and her conundrum. This lady is having an issue with a flashlight. This is the most simple version of the scientific method you could think of. This lady is having an issue with her flashlight. It's just not working. So her observation is, my flashlight is not working. So she gets to thinking she could do some background research. Remember, you could look and see what's known about maybe flashlights not working. But in this case, probably so straightforward. Her question is simply, why isn't the flashlight working? So she has to formulate some hypotheses, right? Remember what the hypothesis is? It's a stated answer to the question. The question is, why isn't the flashlight working? So what's a good hypothesis? So a good hypothesis, again, is the answer to this question. Why isn't the flashlight working? Hypothesis 1. Well, what if the batteries are dead? The batteries are dead. That's a good hypothesis, right? Can we test that? Yeah, we can test it. Can we falsify that? Remember what falsify means? That means if we test this hypothesis and it doesn't work out, we could rule out the hypothesis, like eliminate the hypothesis. Yeah, we could falsify that. Here's another hypothesis she came up with. The bulb is burned out. Okay, is that a good hypothesis? Yes, because you could test it, sure. And you could falsify it. You could rule it out if it's not the bulb. Okay, so let's get started. Let's look at what she does. Let's look at the first hypothesis on the left. She's testing for dead batteries, okay. The prediction would be what? replacing the batteries will fix the problem. What does she do? She pops out the old batteries, she puts in fresh batteries, she then tests the hypothesis, and lo and behold, that did not solve her issue. It did not fix the problem. She put in fresh batteries and still nothing. So she has effectively falsified the hypothesis. She has shown that the hypothesis is not true. So she's basically ruled out the dead battery hypothesis. Okay, now that was an experiment, right? Changing out the batteries was the experiment. A good scientist would change one variable at a time, and the variable being the batteries here. So a good scientist, in case you're curious, a good scientist would put the old batteries back in before testing hypothesis 2, but she may or may not have done that, okay? Because you don't want to test multiple variables at the same time. So what does she do? She hops to hypothesis number 2. The light's burned out, right? Okay, so it's a burned out bulb. So the prediction here would be replacing the bulb will fix the problem. She does the experiment. She gets a fresh bulb. She replaces the flashlight with or the bulb with a fresh bulb on the flashlight. She turns it on. Lo and behold, it works great. Okay, so what has she done here? Did she falsify the hypothesis? No. This is where it gets a little confusing. She failed to falsify the hypothesis, which is actually what you want to end up doing. That shows that you've shown that the hypothesis, in this case, burned out bulb, was the correct hypothesis. It answered your question, and lo and behold, you've solved your problem. You've answered your question. Your observation is now that the bulb was burned out, and putting in a new bulb fixed the problem. And you've learned something about the universe. Okay, so again, why is it called failing to falsify the hypothesis? Let's talk about that for a minute. As a scientist, you want to be your own worst critic, right? You want to make sure that if you're wrong, you figure out you're wrong before someone else does. Okay, scientists do not want to be wrong. And so when you make a hypothesis, which is a stated answer for a particular question, what you as a scientist, if you're a good scientist, what you want to do is try to disprove yourself, okay, the best you can. You really want to try to falsify your hypothesis. You really, really want to try to show that you're wrong. Why? Because if you, despite your best efforts, could not prove that or disprove that your hypothesis is wrong, then it must be right? Or at least you have a good chance of that hypothesis really being the correct hypothesis. Again, what you want to do is actually answer the question. You don't want to come up with your idea of what's answering the question. So you want to attack the hypothesis. You want to falsify the hypothesis. And if you fail, well, guess what? Good job. You failed to falsify the hypothesis, which means that the hypothesis really was the answer to your question. So that's why it's called failing to falsify the hypothesis, because what a good researcher is trying to do is falsify the hypothesis, if that makes any sense. So let's look at another example here of the scientific method. Look here. This is the stream at Richland campus. We got a little stream. We got a couple bridges, right? Bridges that span the stream here. And if you've ever walked across these bridges on campus and looked down, you would notice things like turtles. And the turtles seem to be gathering right near the bridge, right? Right near the bridge, you get this high concentration of turtles. And then as you go further and further away from the bridge, you'll notice that there are fewer and fewer turtles, or less concentration of turtles, less density of turtles. So wouldn't that be an observation, right? Wouldn't that be an observation? Let's go back to this. You've made an observation about the universe, right? That there are turtles in the stream, and they seem to gravitate toward the bridges. Now, what could you do at this point? It looks like you've started a scientific study. You could do some background research. This will allow you to determine what other people have looked at or studied in this field, if that question has already been answered. You could formulate a question. Well, what would your question be? What would your question be? Well, how about, why are the turtles gathering near the bridge? Okay, you might want to spend some time determining if there really is a higher concentration of turtles by the bridge. But let's say you do that, and yes, there are higher concentration of turtles by the bridge. Well, then you would say, why? Why are there so many turtles by the bridge? There's your question right there, right? And then from there you could formulate some hypotheses, which can allow you to make predictions and make experiments. So let's talk about that. So what are some think about this. What are some hypotheses for the question, why are turtles gathering near the bridges? Well, how about because the students are feeding them, right? Think about that. What's happening? All day long, students cross and faculty cross and staff cross the bridge, right? All day long. And have you ever noticed that students they love the turtles and they want to feed the turtles? They'll throw some of their Subway sandwich or some of their bread or crackers or whatnot. They'll throw down some little treats for the turtles. Well, that could possibly draw the turtles by the bridge. So think about it. Is that a good hypothesis? Yes, because we could test it. We could ask the students, I don't know, maybe not to feed the turtles. We could ask them to feed extra. There are multiple bridges. Maybe we could have. You see how I'm making predictions right now? A good hypothesis allows you to make predictions. It's testable. It's falsifiable. So the feeding hypothesis is actually a good one. You know why? Because I could test it by, let's say, on one bridge I tell students to feed. On another bridge I tell students not to feed. I could falsify it because if the feeding bridge and the non-feeding bridge over time have no difference in the number of turtles by them, then it's probably has nothing to do with the feeding, why the turtles are gathering, right? So you see, I'm making predictions. I could test it, I could falsify it. That's a good hypothesis. That's a good hypothesis right there. Now, again, we have multiple bridges, and what you wouldn't want to do, for example, is just tell on one bridge allow feeding, and on the other bridge, what you wouldn't want to do is something like, I don't know, block off the bridge so students can't cross at all. You know why? Do you remember why? Because then you'd be testing two variables at the same time, right? So think about that. You'd be testing two variables at the same time. The students even crossing the bridge is a variable. The students being present on the bridge is a variable. Feeding is another variable. You see that? And when you tell students you can't cross the bridge, you're actually stopping two variables. You're manipulating two variables. You're manipulating the variable of students on the bridge, and you're manipulating the variable of feeding. So there's just an example of a good experiment. All right. Good experiments versus bad experiments. Good experiments test one variable at a time. Bad experiments test more than one variable at a time. So again, why would that matter? Well, what if the turtles are just attracted to the sights and sounds and commotion of students, right? They're curious about it, and that's why they're going near the bridge. Well, in that case, you'd mess up your results by manipulating more than one variable. Okay. So again, a good experiment we could set up on one bridge. We could just leave it alone, or we could promote feeding. We could tell students to feed. We could actually tell them feed more. On another bridge, we could tell the students, well, you can cross the bridge, but please, feeding is prohibited. And we could see, well, what happens with turtle concentrations over time. We could measure it over a span of a week or 2 weeks or a month and see what happens. Okay, well, let's say we do that experiment and there's no change in number of turtles by the feeding bridge and by the non-feeding bridge. Well, then what would we do? We've now falsified the hypothesis, which means we've defenestrated the hypothesis. We've thrown it out the window, and we can get started with a new hypothesis, right? We basically come up with another hypothesis and test that one. So for example, what if it's a hot day and you see under the bridge it's all shady? What if the bridges are just providing shade, and the turtles are gathering near the bridges because of the shade? That's another hypothesis. Is it a good one? Sure, we could test it. For example, we could put shade. We could hang shade somewhere else. We could hang a giant 2x4, not 2x4. I'm sorry, plywood, a giant square piece of plywood somewhere else, which provides a lot of shade. and see if they go toward that shade. We could put heat lamps under the existing bridges that make it uncomfortable and don't provide any relief from the heat. I don't know. We could do a number of things is what I'm saying. We could test that. We could falsify that. And let's say we test it and that does affect the turtle population or turtle density. Well, then we've failed to falsify the hypothesis. That means shade really was the reason, the answer to our question, and we can move on. So that is a good scientific study. What you should realize though about science is that failing to falsify a hypothesis does not prove that hypothesis. Okay, you don't really prove anything. And good scientists, by the way, rarely, if ever, use the word proved. Why? Because there's always a chance that you're wrong, and there's always a chance that you've overlooked something. So a good scientist would not say, hey, guess what, I proved X, Y, and Z, or I proved turtles are by the bridge because of I don't know, because of the shade. They would say, my data suggests that shade affects turtle migration, turtle behavior, turtle gathering. My results provide strong evidence for this. They wouldn't say things like, oh, I proved this. Well, because if you say something like you proved X, Y, and Z, then you're most likely going to be wrong at some point, and that doesn't look right, because a scientist is not trying to be wrong. A scientist is trying to show with evidence data, and the data can never be 100%. You know what I mean? So for example, let me give you an example. Let's say this lady, she changes out the bulb, and she's so excited. She failed to falsify the hypothesis. The flashlight worked great, and she goes around telling her colleagues that, guess what? I proved that my bulb was burned out. Okay, remember the hypothesis was burned out bulb, right? She was so excited, she says, my bulb is burned out. Okay, well, what's the problem with that, right? What if there's someone in the audience raises their hand and says, excuse me, but did you test the old bulb once you took it out of the flashlight? Did you actually, attach it to another flashlight, or did you test the filament with some kind of tester, bulb tester? And what if she didn't, right? So her whole hypothesis was the previous bulb was burned out, right? And what's the one thing she didn't do? She didn't test whether the bulb was burned out. Do you see the problem there? Yeah, now you look like a clown, right? You look like you don't know what you're doing. You look like someone who, made statements that they didn't really back up. And it would have been much better for her to say, well, from what my studies, from what I did, my evidence suggested that the previous bulb was burned out. This way, when someone raises their hand and says, oh, but did you try this? It's like, oh, I haven't, but the main thing I was trying to focus on was resolving the issue and not really testing the old bulb. However, we should have probably done that. And thank you for your input. Maybe I'll do that next time. You see, that's a lot better than saying, I proved the bulb was burned out, when you did everything but actually test that bulb. Okay, so anyway, I hope this makes it clear. Good scientists rarely, if ever, use the word proved. Instead, they'll say, we've shown strong evidence for, etc., etc. And then remember, what was the last step of the scientific method? Communicate your results to others. Well, this is called reporting scientific work. So there are these journals. You can think of them as magazines called scientific magazines, and in them you publish scientific studies. You may have heard of journals called Science, the journal Nature, the journal Cell. These are very prestigious journals where scientists, they submit their findings. Okay. And the good part about these journals is they don't just take your word for it. It's like, oh, hey, Dr. Jones, Dr. Smith, we know you're a famous scientist. We're just going to take your word for everything, and we're just going to publish it right away. No, what these journals do is they send those results. They send those manuscripts. The manuscripts are the typed up results with all of the abstract, which is basically a concise summary, the introduction, which is a background, a background to the field, materials and methods, so other scientists can reproduce your results. What were your results, and a discussion of those results. Right, this whole thing is called the manuscript. The manuscript that you submit to a journal, the journal staff sends your manuscript to other scientists in the field, right? There could be a scientist in Australia. There could be 2 scientists in America. There could be a scientist in, China. And all of these different scientists will sit down, read your manuscript. Let's say our turtle study manuscript. They'll read your manuscript, and they will determine whether it has merit. Like, did you do good experiments? Did you have good experimental setup? Did you test all of your variables? Did you test only one variable at a time? Did you do good statistics? Did you count enough turtles? Or was there, if there were only 2 turtles in the whole stream, that's not a good study. You need dozens of turtles, right? At least. So was it a good study? And if it wasn't a good study, they could suggest revisions. They could say, Dr. D, you should really try doing this experiment with more turtles. So you should go somewhere where there are more turtles. 2 turtles is not enough to do this experiment. So come back when you've looked at, 100 turtles. Well, then I would get that feedback from the journal. The journal would say, hey, Dr. D, that was a good study. However, your peers have suggested the following revisions. Please make the following revisions. And then, so that makes my science stronger. I need to go back. I need to do a better study. And then once I've done that, I could resubmit, and hopefully the peers would be satisfied, and they will publish my manuscript in the journal. And then I could get some notoriety for that, and I could contribute to knowledge and contribute to science that way. That's what peer review means, and it's how science works. And guess what? I'm not done. At that point, even if they were able to publish my manuscript, I'm not done. Other scientists need to be able to look at my materials and methods, and those scientists need to be able to redo those experiments and recapitulate my results. They need to be able to reproduce my results. And if they can't reproduce my results and see the same thing when they do the shade difference and they see the turtles migrating in the way I saw it, well, then pretty soon people will realize that my results aren't very trustworthy and that I don't do good science. And then I'm ridiculed out of the field. People understand that my science isn't very good. So you can't just put any old stuff out there if you're a scientist. You need to, again, you're the one who is attacking your own hypothesis and trying to falsify your own hypothesis the most. Okay, and you're the one trying to disprove yourself. Okay, you can't prove anything, but you can disprove stuff all day long. All right, so that's how science works. And so what have we done so far? Let me flip to the beginning real quick. Again, what was biology? Biology was the scientific study of life. Okay, we have just discussed what is a scientific study. Okay, and now we're going to discuss what is life. So I'm going to take a brief break here, and you could take a break, hit pause button, and maybe go to a restroom, grab a drink, or stretch your legs, and I'll explain what is life. Hey everyone, welcome back. Hope you had a nice little break there. One slide that I wanted to get to before we start talking about life is this one here. It covers theories, scientific theories. What you need to understand about science is a theory is a tested and confirmed explanation for observations or phenomena. What this means is that there's a huge body of interconnected evidence, huge number of manuscripts published in reputable journals that supports each different theory. So a theory is something that's all but proven. Okay, compare that to the general meaning of theory. The general meaning of theory means it's like a guess. It's like if your slippers go missing. I have a theory my dog took it. That's what theory means in general, in layman's terms. But to a scientist, remember when I said a scientist will not use the word proved? Okay. Well, the closest you can get a scientist to saying they've proven something is they say it's a theory. Okay, so just be aware there are the Big Bang theory, the theory of evolution, the general relativity theory, cell theory. All of these are theories. All of these are theories. Gravity is a theory. All right, so a theory is tested and confirmed, and there is no significant supporting evidence to the contrary. Okay, so just be aware of that. Theory to a scientist means a whole different thing than a theory to someone just in everyday life. Okay, so that being said, we'll move on to what is life. Remember, biology is the scientific study of life. So what is life? Life, unfortunately for us, it defies a simple explanation. There's no really super easy definition. And there's creatures on Earth that some scientists would argue are alive, other scientists argue are not alive. So it's not so clear cut and well defined. So what scientists agree on is that there are certain characteristics of life. These are the characteristics: order, sensitivity, or responding to environment, reproduction, adaptation, growth, development, regulation, homeostasis, which is a fancy way of saying being able to maintain internal conditions, ideal internal conditions, energy processing, the ability to evolve. These are the things, these are the characteristics of life. And if you're a living thing, typically you possess most, if not all, of these characteristics. So humans possess all of these characteristics. Some living things that do not possess each and every characteristics, but they'll possess most of them. Okay, so as long as you possess most of these characteristics, you can be considered a living entity. Okay, so that's, I guess, the best definition of life. Now, when you're studying biology, you're obviously studying organisms, right? You know what an organism is at this level here. An organism is a creature. You and I are the human organism, right? And you can break that down into simpler levels, or you could go up into more and more complex levels. So just to give you an idea, we are an organism, but you could break us up into our different organ systems. For example, we have the nervous system, the cardiovascular system, the immune system. And each one of those has different organs which are involved, which include tissues. Tissues are parts of the organs. These tissues are made up of individual cells. In fact, if you're not a multicellular organism like a human, if you're, for example, a single cell organism like a bacteria, well, then you don't actually possess tissues, organs, organ systems, or anything like that. You are a cell, and that's it, one cell big. However, we are multicellular organisms, so our cells make up our tissues make up our organs make up our organ systems, and those organ systems together make us up, which is the organism. And of course, cells are made up of organelles. If you're wondering what is an organelle, organelles, you can think of them as functional parts of the cell. Okay, organelles are functional parts of the cell. So what do I mean? Have you heard of the nucleus? Okay, that's a part of the cell. Have you heard of the mitochondria? Right, that's a part of the cell. Have you heard of ribosomes? That's an organelle, a part of the cell. So if you haven't heard of those things, don't worry about it. But just be aware that just like us organisms are made up of organs and organ systems, cells are made up of organelles, functional parts of the cell. And those organelles, of course, are made up of molecules, which are made up of the atoms, which are essentially the elements. We're going to be talking all about that in Chapter 2 and Chapter 3. Okay, now that gives rise to the organism. But we could go in more and more broad here. A group of the same species of organism is called a population. So, for example, at Richland campus, you would have the human population. You could have the turtle population, fire ant population, crepe myrtle tree population. Populations are groups of the same species in an area. Okay, so that's what you need to be aware of. What is a community? A community, to a biologist, a community means all of the different species in an area. So on Richland, you have the population, the human population, the turtle population, right? All those populations together make up the community. So for example, the Richland community would be all the humans, all of the turtles, all of the ants, all of the trees and bushes and fish and all of the birds, etc. and grasses and bushes and all that. So what's the ecosystem? You can think of ecosystem as community plus environment. Now you're not just talking about the community, which is all the different creatures on Richland campus. You're talking about the dirt, the air, you're talking about the stream, all of that. That's the ecosystem. And of course, all the ecosystems put together make up the biosphere. Bio means life and sphere meaning it's a big circle. So we're talking about the big circle of the Earth. Right, the entire planet is the biosphere. It's made up of ecosystems, which are made up of communities, which are made up of populations. You get the point. All right, so like I said, there are different organisms on the planet, different species. So what is a species? A species is a group of organisms. If you're talking about multicellular species, it's a group of organisms that can interbreed and have fertile offspring. Okay, so for example, our species is called sapiens. That's our species. Homo is our genus. Genus means even another group of closely related organisms. So Homo is our genus. It's a group of closely related organisms. Sapiens is our species. It is the population. It is the group of organisms that can interbreed and have fertile offspring. What do I mean by fertile offspring? So for example, when humans breed, they have offspring that can then have more humans, right? But let's say members of a different species in a genus, a different species, let's say a horse and a donkey breed. They can actually breed. A horse can breed with a donkey. However, their offspring is not fertile. It's called a mule. The offspring is called a mule, and a mule is not fertile. A mule cannot have more mules, right? Same with a lion and a tiger. They make ligers. Ligers are not fertile. They are not fertile offspring, okay? That's why they are different species. They are not the same species, okay? And this two-word epithet where you got genus and species, this is called binomial nomenclature here. It was derived by or brought to you by Carl Linnaeus, the Swedish botanist, 18th century. And there's a very specific way of writing this binomial nomenclature. The first word is, again, the genus. The second word is the species. The first word, notice that it's capitalized, in this case, capital H. The second word has to be lowercase. All right, so that's very important. Capital, lowercase, and then the whole thing needs to be either underlined or italicized. Does that make sense? The whole thing is either underlined or italicized for it to be correct binomial nomenclature. Okay, so that's binomial nomenclature. And the study of how organisms are related to one another, this is the study of taxonomy. So a taxonomist is someone who studies different organisms on the planet and studies how closely related they are, puts them into species, puts them into genus, and then order, and all the other classifications. Let me talk to you about that. Actually, let me talk to you about that here. A taxonomist would take a creature such as the black bear. Okay, this is Ursus americanus, the black bear, and the black bear is a species Americanus, but it is in the genus Ursus. Notice Ursus is capitalized. Ursus would be all the other bears, right? So you've got the black bear and then different species of bear. You've got the polar bear and you've got the brown bear, the grizzly bear, and they're all in the genus Ursus, but they are of different species, okay? Then you have different layers. You have even more layers, and they get more and more broad, more and more less and less general. More and more general. I should say. You've got family, which includes, in this case, a lemur-like creature. You've got order, which includes, in this case, all of the carnivores. You've got class, which includes all the mammals. You've got phylum. Notice how we're getting more and more broad, more and more broad. A phylum, which includes all of the chordata. Chordata means you've got a backbone. You've got kingdom, which includes all of the animals. You've got domain, and domain is eukarya. So I'm going to tell you what eukarya means in a little bit, okay? But again, a taxonomist would say, okay, here's a new species of black bear, for example. Where does it fall into what kingdom, in what phylum, in what class? And so it's good to understand this whole taxonomic hierarchy. This is called the taxonomic hierarchy, from species all the way to domain. Okay. In fact, you may want to memorize this. Domain, kingdom, phylum, class, order, family, genus, species. And I've got a nice little video here you can click on, bottom left, which will help you to memorize this. I've got a nice little mnemonic device for you. You may want to watch this video here. Okay. So that's taxonomy. Let's go back and talk about the different types of cells. There are two types of cells that you need to know about. There are eukaryotic cells. These are cells that have membrane-bound organelles. The main membrane-bound organelle is the nucleus. And then you've got prokaryotic cells, which are simpler cells. They do not have membrane-bound organelles, and they do not have a nucleus. So again, all cells have a membrane around the cell. That's called the plasma membrane, okay? Again, you should understand that all cells have a membrane around the cell. That's called the plasma membrane. All right, the difference between eukaryotic cells and prokaryotic cells, the main difference is that despite the fact that all cells have this plasma membrane, eukaryotic cells have more organelles inside which have membranes, whereas prokaryotes do not have organelles inside the cell that have membranes. They have organelles, don't get me wrong. They just don't have membrane-bound organelles, okay? So the eukaryotic cells have a nucleus. That's the main membrane-bound organelle. But they also have, for example, they have mitochondria, which is another membrane-bound organelle. Plants have chloroplasts, which is another membrane-bound organelle. You can have endoplasmic reticulum, which is another membrane-bound organelle. You don't need to know about these structures now, but I'm just listing off some. The Golgi apparatus is another membrane-bound organelle. None of the prokaryotic cells have these things. Prokaryotic cells have non-membrane-bound organelles. For example, the ribosomes, right? Ribosomes are non-membrane-bound organelles, okay? So you and I, we are eukaryotes. What does that mean? Our cells, you know, we are made up of billions of cells, right? Our cells are eukaryotic cells. If you were to crack open one of our skin cells, for example, you would see inside a bunch of membrane-bound organelles. And of course, again, what's the main membrane-bound organelle? The nucleus, right? But a prokaryotic cell or a prokaryote, for example, E. coli or Salmonella, these guys don't have a nucleus inside. So if you cracked an E. coli open, you wouldn't find a nucleus inside or any other membrane-bound organelles, okay? So here's an example of a eukaryotic cell, okay? This would be, for example, your cell. And here's an example up here at the top right of a prokaryotic cell, okay? Notice the eukaryotic cell has all these big structures inside. These are membrane-bound organelles. And you see the big blue one in the middle? That's the nucleus, okay? The prokaryotic cell, notice how it doesn't have membrane-bound organelles inside. Now, here's something you need to understand. Okay, let's take a minute to think about this. Okay, it's actually kind of important to understand. I said all cells have a membrane around the whole cell. That's called the plasma membrane. But only the eukaryotic cells have membrane-bound organelles inside, including the nucleus. So what's this blue stuff in here? Look at the prokaryotic cell. What's this blue stuff in there? Okay, so what you need to understand is all cells have DNA inside, right? So our DNA is inside of our cells, right? So what's the difference between a nucleus and not, okay? In eukaryotes like you and me, our cells, like our skin cell, has DNA, but that DNA is inside of a membrane, okay? Does that make sense? Our DNA is all in, our chromosomes are inside of this membrane. It's actually a double membrane. We'll get into that later. but that's what the nucleus is. It means that your DNA is inside of a membrane. Okay, that's nucleus. The E. coli, look at the E. coli here at the top right. For example, let's pretend this is E. coli. It also has DNA. The blue stuff's DNA. It's its chromosome. Okay, it has DNA. Okay, but its DNA is just floating around in the cell. All right, the DNA is not inside of a membrane, so that's why we don't call it a nucleus. You see here, the DNA is just floating around the cell. So the DNA is not a nucleus. In this case, you would call it a nucleoid. A nucleoid. All right. Now, again, eukaryotes have the DNA inside of a nucleus. Prokaryotes have the DNA floating around in the cell. By the way, you see this fluid inside of the cell, just this fluid inside of the cell? That's called the cytoplasm or cytosol. All right. In the eukaryotes, our DNA is inside of a nucleus, inside of a membrane, and so the DNA is not in the cytoplasm, is it? It's inside of the nucleus. In a prokaryote, however, look what's going on. The DNA is floating around in the cytoplasm. Okay, so good to understand. All right. So now we know there are two types of cells. There are eukaryotic cells and prokaryotic cells. What you need to now understand is that there are three major groups of life, all right? Look, bacteria, archaea, and eukarya. These are the three major groups of life, and all living things, all living creatures fall under this group, bacteria, this group, archaea, or this group, eukarya, all right? Two of these groups are prokaryotic. Bacteria and archaea, these creatures are prokaryotic creatures, and they are single-cell creatures. This group, eukarya, to which we belong, by the way, you and I belong to this group, eukarya, these are all of the creatures that are made up of eukaryotic cells, okay? And you and I are a multicellular creature, right? We are humans. But you should understand that there are some eukarya that are single cell. Have you ever heard of, for example, amoeba or protozoa or paramecium? These are single cell eukarya. So eukarya can be single cell creatures. They could be multi-cell creatures. However, archaea and bacteria, which are prokaryotic, these two major groups of life, these are single cell creatures. Okay, now here's the thing. These groups have names. They're called domains. Okay, there's the domain bacteria, the domain archaea, and the domain eukarya. So let me show you. Do you remember this? You got the black bear, and look what domain it belongs to. Remember, there are three domains of life? The black bear belongs to eukarya. Where would we belong? Which domain? We would also be in eukarya. Okay, does that make sense? We are also in eukarya. All right. So there are three domains of life: eukarya, archaea, bacteria. Again, the domains bacteria and archaea, they're composed of single-cell creatures. Okay, and eukaryotes can be single or multicellular. In fact, the eukarya you may be familiar with. They include the plants, the fungi, animals, and also creatures called protists. You know what protists are? Protists, a lot of them are single-cell creatures. These are the ones I was telling you about. These are the protozoa. These are the ones that are paramecium or trypanosoma, amoeba. A lot of these are single-cell eukaryotes. Not all of them, but a lot of them are. Algae and things like that fall under protists. Okay. So you can see plants, animals, fungi, protists. These are all types of eukaryotes. Okay. And that's it. That actually wraps up this chapter. The last concept here is you should know again. Remember, there are three domains: bacteria, archaea, and eukarya. Remember, these are the three major domains of life, and all living things fall under one of these three domains. And what you should realize is bacteria are prokaryotic, archaea are prokaryotic, eukarya are eukaryotic. However, look at this. Okay, this is what you need to understand. Look at this. This is called the phylogenetic tree. This is the tree of life, okay? And how it shows you how closely different organisms are related to one another. Notice this. there's a common ancestor here in history. There's a common ancestor, and that common ancestor branches off two directions. One becomes bacteria, the other becomes the archaea and the eukarya. You know what this means? Do you know what this means? This is actually kind of interesting if you think about it. What this suggests is that eukarya, which includes you and me, and archaea, which are these creatures that are prokaryotes that usually live in extreme environments, even though archaea and eukarya, are really different, one's prokaryotic, one's eukaryotic, we share a more common ancestor than archaea does to bacteria. Okay, again, what does that mean? In layman's terms, that means that archaea are more closely related to us eukarya than they are to other bacteria, to other prokaryotes called bacteria. So these prokaryotes are more closely related to us than they are to bacteria, which is really fascinating to think about. We have a more common ancestor with archaea than bacteria do. Okay, so that wraps up chapter 1. Again, welcome to Biology 1406. I hope this is a good semester for you. I've got these videos here. By the way, you see, whenever in the notes, whenever you see this at the bottom left, you see Dr. D, click me. You can click that icon and you'll get another video that will help you to understand the concepts better. So go ahead and click on those and you'll get mini videos that you can watch to help clarify some of these points. Okay, I'll try to put these in, increase the number of these as I can and make more videos as I can. Okay, thank you very much for watching, and tune in next time.